Next, we are going to see about how the plasmid replication is going to be taken place. As we said, a replication origin is going to be present. The plasmid replication to be taken place by bidirectional replication. It is also known as theta replication or Karen's replication, because a bidirectional replication was first found to be discovered by Karen's. So it is also known as Karen's replication. So it was first visualized by John Cairns in 1963 in the chromosomes of plasmid. So she found it through autoradiographical studies. First, a plasmid DNA is going to be present as a double-stranded circular DNA. It has a origin for the replication, where the replication is going to be started off. So it was present inside the cell, that is bacterial cell. He chose a E. coli cell. He chose up the E. coli cell and grew it in the medium, as the bacteria can grow in certain medium. So in the lab she grew it in the medium, which is going to consist of tritium labeled, that is radioactively labeled tritium. A tritium is labeled so that when the bacterial cell is going to be grown in this medium, it can take tritium labeled thymidine containing medium. As we said here, tritium labeled thymidine containing medium. So when the E. coli cells grown, it needs a supply of thymidine. When it needs the supply of thymidine, it takes up the tritium labeled thymidine into its site for the replication. So after several periods and times, he isolated that E. coli cell and lysed the cells without breaking of the chromosomes. So no chromosomes are break up. Only the lysed cells can be taken up. So the chromosomes is collected first. Next, a collected chromosomes are going to be filtered off to get rid of other materials, other biomolecules. So chromosomes are collected in purified manner. Then these chromosomes are going to be placed on the glass slide coated with emulsion sensitive to beta particle, which means a tritium is going to be labeled in thymidine cells. So the tritium labeled thymidine cells are going to convert it into normal hydrogen. Tritium is going to be a heavy hydrogen, where normal hydrogen is going to have a 1 valence. So tritium is going to be 3. So it undergoes a decay, which causes it to release the beta particles, and it can convert a tritium to normal hydrogen with the release of beta particles. So the region where the beta particles get released, which lead to a radioactive decay. So he photographed by autoradiography technique and find out the replication. From this replication, where she saw that in the photograph there was 2 forks to be present at the central region, which means where the replication to be taken place by in the two region in the opposite direction. Both the strands are going to be replicated in the opposite direction, where it taken place simultaneously. Both unwounding and replication taken place simultaneously. So the coordination process is going to be taken place during the bidirectional replication or theta replication. Why it was known as theta replication means, here we can see this is a circular DNA of plasmid. It's going to have a origin of replication here. So when the replication is going to be taken place, a DNA is going to form a both leading strand and lagging strand. Synthesis both leading strand and lagging strand, where as one of the DNA is going to synthesis in this direction, another is going to be in this direction. So the direction is going to be in the opposite manner and lead to synthesis. Here we can see it was going to synthesis in this direction, another strand is going to synthesis in this direction, the opposite directions. So during this photographed, it was shown to be found like a Greek letter theta. So this replication, that is bidirectional replication, is going to be also known as Karen's replication or theta replication due to its resemblance as theta. So in this manner, after they replicated at the end of germination site, it forms a catenance. Like this, a catenance can be formed at the end of the replication, two plasmids. In this, one of the plasmid is going to undergo the semi-conservative mode of replication, which means one of the plasmid is going to be a parent, another is going to be a daughter plasmid. Each of the plasmid to be formed is going to consist of both parent and daughter strands. It is known as semi-conservative mode of replication. The end of a region where both of the plasmid get connected is known as catenance. These catenance are going to be released. These catenance, the site is going to release these two newly formed plasmids. So it was released by the enzyme topoisomerase. A topoisomerase is going to create a supercoiling of these two plasmids and lead to release of these two newly formed plasmids. So here we can found both the unwounding and the replication is going to be taken place in the cooperative manner or coordinated manner. That's what she visualized in these studies. This is the bidirectional replication of plasmid. So next we are going to see about the function of plasmid. Plasmid is going to be used in many ways in genetic therapies and genetic technologies. It is used as a vector. Vector is a molecular vehicle, which means it can carry a gene of interest during the recombination technology. It can helpful in production of rDNA, that means recombinant DNA. They are commonly used for cloning during the genetic technologies and amplify the particular gene. So in the plasmid, a region where can be cut, a restriction site is going to cut the gene and the gene of interest going to be integrated into it, and the plasmid is going to be introduced into the bacterial cell. After a certain time, it going to produce a many plasmids by its autonomous replication so that it can amplify the particular gene. Therefore, a common plasmids to be used as a vector are RP4 plasmid of Pseudomonas, whole E1 of E. coli, and Y2P of yeast. Yeast can also consist of plasmid. Next, we are going to see about the another important function, that is protein production. As we said before, a gene of interest going to be introduced into the plasmid and it can produce more clones of genes. When the more clones of genes is produced, a gene can express its, if a gene like insulin gene is going to be integrated into the plasmid, it can be introduced into the cells, bacterial cells, which lead to amplification of the gene. After the amplification, a gene, that is insulin gene, integrated into a plasmid can express its function. That means it can produce the insulin protein, insulin hormone. So the insulin hormone can be harvested through this technique. So it can be used as a expression vectors. As we said before, it can be used as a vector, it can be used as an expression vector, so that a insulin or a gene that can produce certain proteins can also be harvested. So it can be used as a microscopic factories for the harvesting of specific protein. Next, it can be used in gene therapy, which means if a person is going to lack a certain enzyme due to inborn error of metabolism, a lack of enzyme which lead to accumulation of enzymes product and lead to many metabolic disorders. So the enzyme can be synthesized through gene therapy, which means a gene of interest, a gene that lacks the production of enzyme, lacks in the person for the production of enzyme, are going to be introduced in this plasmid, and it was introduced into the human cell, into the bacterial cells, and introduced into the human cell, which lead to production of amplification of a gene and production of the enzyme that is of interest, which is used in gene therapy. That means it can be used in production of therapeutic gene. So it can treat the genetic disorders. So a plasmid can also be used in genetic disorders in genetic technology, protein production and vector. These are the uses of plasmids. Thank you.